Bismillahirrahmanirrahim. Today we are going to learn about the physiology of memory. Memory is the ability to store new information and the ability to refresh the stored information. There is always a confusion between the memory and learning. Learning is the ability to acquire new information, while memory is the ability to store the new information. Memory is stored throughout the brain, but four major parts are there which are responsible majorly for the storage of memory, for the storage of new information. I'm going to write them. Number one, the cerebellum. Number two, the amygdala. Number three, the hippocampus. And number four, the prefrontal cortex. Going to discuss each and every one in detail. Going to the first slide. This is the brain. This part represents the forebrain. This part represents the midbrain, while this part represents the hindbrain. This is the frontal lobe of the cerebral cortex. This is the parietal lobe. This is the occipital lobe, and this is the temporal lobe. Coming towards the cerebellum, this is the cerebellum. This is the part of hindbrain. This is responsible for the storage of memory regarding the procedure. This is responsible for the storage of procedural memory. I'm going to write it. The cerebellum is responsible for the storage of procedural memory. What is procedural memory? Procedural memory is the memory which has something to do with the procedure, which has something to do with the procedure and balance also. I'm going to show you the example. Look at this example. To play the piano is a type of procedural memory. The information regarding the procedure of playing the piano is stored in your cerebellum. So cerebellum is responsible for the procedural memory of playing the piano. Similarly, look at this example. Cycling is the best example of the procedural memory. The information regarding cycling is stored in your cerebellum. Similarly, you may have seen a lot of birds, a lot of animals are there uh, which maintain their balance by sitting on the twig of trees. So this is stored in their cerebellum. So the cerebellum has maintained the balance, and that balance are maintaining the balance, or the memory of maintaining the balance is stored in their cerebellum. Similarly, a lot of examples are there which deal with the procedural memory. For example, the driving, for example, the swimming, for example, the typing. All of these information are stored in the cerebellum, in this part of the hindbrain. So if we remove the cerebellum from an expert driver, that driver will no more be a driver. After removing the cerebellum, he will no more be able to drive the car. He will no more be able to drive. Similarly, the acrobat has also very well developed cerebellum, because a lot of memory regarding the balance maintaining, the maintenance of balance has been stored in the cerebellum of the acrobats. Now coming towards the second part, that is the amygdala. This is the amygdala, and this is located in the temporal lobe. In the middle region of the temporal lobe, this amygdala is located, and this is the almond-shaped structure. And amygdala is responsible for the storage of a special type of memory that is called fair memory. I'm going to write it. The fair memory. Fair memory is the memory regarding the fear, regarding the fright, regarding the stressful condition, regarding the when someone is in fair condition, in fright condition, in stressful condition, this amygdala got active, and this by getting active, this amygdala, it stored the information regarding the fear, regarding that condition. I'm going to show you an example which will demonstrate the function of amygdala. This is mice, and this mice is placed on the flat. This flat is not having any sort of irritation, any current, or any foot shock, etc. And this mice is active. This mice is not showing any fair response. Now, if we apply the foot shock to the mice, this mice will get freezed. But during applying the shock to the foot of this mice, to the path of this mice, what will happen? During applying the shock, we also apply the bill. So this mice get the information of shock and bill at the same time, and it saves the information of bill or listening to the bill and getting the foot shock in their amygdala. So this mice store the information of listening to the bill and foot shock in its amygdala. And this mice is showing a freezing response after listening to the bill and applying the shock. Now, if we do not apply any shock after so many repetition of this procedure, or after so many repetition of this experiment, this mice is applied with only bill. After listening to the bill, this mice will get freezed. Though there is no current, there is no foot shock, still the mice get freezed. Why the mice get freezed? Because the information regarding the fear is stored in its amygdala. So after listening to this bill, this might assume that he is in fair condition. This might assume that he is getting the foot shock, but there is no foot shock at all. Still, this mice is showing the freezing response. Why this mice is showing the freezing response? Because this mice has stored the information of fear regarding the foot shock in its amygdala. So after listening to this bill, he assumed himself in danger in stressful conditions, so he showed the freezing response. Now, if we remove the amygdala from this mice and give foot shock time and again, give foot shock to this mice and apply the bill also. Then, after applying the bill to this mice, he will not show any freezing response because the information regarding the fare cannot be stored in this mice. The information can only be stored when there is amygdala in this mice. So this is the amygdala, and this amygdala is located to over here. This is the frontal plane of the brain, and over here in the temporal lobe, in the middle area of temporal lobe, this amygdala is located. Now coming towards the third part, that is the hippocampus. This lobe shaped structure is the hippocampus. Hippocampus is very important part of the brain, very important part of the memory. It store a special type of information or memory that is called the explicit memory. Explicit memory, that is also called declarative memory, that is also called spatial memory. The explicit memory, spatial memory, or declarative memory is a type of memory which deals with the information regarding the surrounding, which deals the information regarding the space, which deals the information regarding the events. So the hippocampus stored the information regarding special events in the life. This part of the brain remain active during the event, during special events. For example, if you are attending a marriage ceremony, you are attending the funeral, if you are attending the birthday party, if you are getting a picnic party. All of these events are stored in your brain. Similarly, the information regarding the surrounding is also stored in this hippocampus. How the information regarding the surrounding is stored? You know a special type of city, special type of building, you know a special type of street. Now, so because of the structure of that street, because of the structure of that building, so all these information regarding the surrounding and space and pathways are stored in your hippocampus. I'm going to show you an example. This is the simple maze, and this maze we put a mice. This maze is having multiple pathways. If we put some sort of food over here in this section or in any other section, and start teaching this mice how to lead towards this food, how to lead to this food, how to follow this food. this mice will go toward this food. After teaching this mice how to read this food, he will he will have the data regarding the surrounding, regarding the pathways to reach this food. How will he reach this food? Once we teach this mice how to reach this food following multiple pathways inside this maze, then after reaching this food, this mice will store the information regarding the surrounding of this maze, regarding the pathways of this maze. And once again, if we put food in any other part of this maze, for example, if we put the food over here, this mice will be able to learn, this mice will be able to read this food, because he is having the information regarding the full maze. He is having the full data regarding the information regarding this maze, regarding the pathway. So he has stored the information regarding the surrounding. He has stored the information related to the area, related to the surrounding, related to the pathway in his hippocampus. Now now if we remove the hippocampus from this mice, after removing the hippocampus from this mice, this hippocampus removed mice will not be able to lead to any food. This mice will not be able to learn any information regarding the simple maze. A very simple maze cannot be followed by this mice, because this mice will not be able to store any new information regarding the pathways, because there is no hippocampus in the mice. So the hippocampus is responsible for the storage of information regarding the surrounding, regarding the pathways inside the animal, inside the human, inside the many other birds, etc. Now coming towards the very important part of the memory, that is called the prefrontal cortex. This is the prefrontal cortex, and this prefrontal cortex is located inside the frontal lobe of the cerebral cortex, or this is located in the frontal part of the frontal lobe of the cerebral cortex, the prefrontal cortex. And the prefrontal cortex is responsible for the storage of special type of memory that is called semantic memory, semantic tasks and memory. So the semantic tasks and semantic memory is stored inside this prefrontal cortex. What is semantic? Semantic mean anything which is related to the words, which is related to the words. So what is the semantic task? Semantic task is the to listen about any word and to give an information about that word is called semantic task. What is semantic task? For example, I have told you that there are four parts of the cerebellum. There are four parts of the memory: the cerebellum, the amygdala, hippocampus, and prefrontal cortex. So if you listen about the cerebellum, you also know about the spelling of this cerebellum. If someone tell you about the flower, if someone tell you about the flower, you directly in your mind comes FLOWERS, flower. So what is, from where this word come? The memory regarding the words, the memory regarding the words of any language, the memory regarding the words of the Arabic words, or the Chinese words, or the English words, or the Urdu words, or any other words can be stored in your prefrontal cortex. The prefrontal cortex is responsible for the storage of information regarding the word, any topic which we learn, any topic that is, for example, this topic is also regarding the words. I have written a lot of words, and all these words have been saved in your, have been memorized or saved in your prefrontal cortex. The prefrontal cortex is responsible for the storage of semantic memory or semantic task, and these tasks are this memory is regarding the words. So these are the four parts of brain which are responsible for the storage of information regarding the fright, regarding the events and surrounding, and regarding the words. And all of these areas are coordinated by the hippocampus. Now coming towards the molecular basis of memory. If we stored any information inside the brain, inside the cerebellum, inside the hippocampus, inside the amygdala or prefrontal cortex, we give structural basis to that memory, and that structural base of memory, or that anatomical structure of memory, is called what? That is called engram. Engram is the structure inside the brain which store the information. And what is engram? Engram is a small part of the neuron. Engram is the part of the neuron which is responsible for the storage of spatial information. I'm going to show you in detail. This is the structure of neuron. This neuron may be in any part of the memory of the brain. So this may be in the hippocampus, this may be in amygdala or the prefrontal cortex, etc. But how they store the memory? First of all, the information comes through the come in the form of the impulse, and that impulse uh, will lead to the secretion of neurotransmitter, and that neurotransmitter will be secreted by the neuronal ending, and this neuronal ending comes inside the hippocampus. Let's suppose this is the hippocampus. So this neuronal ending will secrete what? Will secrete the glutamate towards the synapse. This synapse is having the postsynaptic neuronal ending also. This is the postsynaptic neuronal ending. So this postsynaptic neuronal ending will contain special type of receptors of glutamate receptor, and those receptors are called what? These are called AMPA and NMDA receptors. Two types of receptors, AMPA and NMDA receptor. NMDA receptor. So these two types of receptors or glutamate are actually the channels of sodium and calcium. So when the neuronal ending over here secrete what? It secrete the neurotransmitter. The neurotransmitter is secreted over here, and this neurotransmitter will combine with the uh, with the with the receptor site over here on this glutamate receptor, NMDA receptor. It will lead to the opening of this channel. This channel will start the influx of sodium and calcium inside this neuron. So the influx of sodium and calcium will lead to the formation of what? It will, first of all, it will create an action potential over here on this neuron. Secondly, the influx of calcium and sodium over here will also lead to the formation of second messenger, and that second messenger is CAMP. So the CAMP formation will occur over here, and those CAMP will do what? That will sensitize the glutamate receptors on this postsynaptic neuronal ending. They will increase the sensitivity of these receptors and also increase the number of receptors on the postsynaptic nerve ending. So it will create a condition over here. The neurotransmitter which are secreted in the synaptic lip, that neurotransmitter will combine with some receptor, and after detaching from the receptor, it will go back towards over here and recycle toward the vesicle, and again that will be secreted over here, and this cycle will be continued. This cycle and this condition only happen in the memory depositing or memory storing area inside the brain. So the phenomena or the condition will occur over here, that is called long-term potentiation. Long-term potentiation, for example, normally in the neurons inside the brain or inside the peripheral nervous system, the impulse or the neurotransmitter when are secreted in the synaptic cleft, that activate the receptor or generate the action potential for a few milliseconds. But inside the memory storing area inside the brain, the action potential and the impulse and the secretion of neurotransmitter is continued for long interval, is continued for many hours, from minutes up to hours, and this phenomena is called long-term potentiation. Long-term potentiation is the very important phenomena memory inside the brain. Long-term potentiation. So the long-term potentiation mean the long-term secretion of what? The long-term secretion of the neurotransmitter and the long-term generation of action potential over here for many hours. So what will it do if the information is very important for our mind? If the information is very important for us, it will depend upon the information to information. If the information are very important for us, it will create the action potential for many hours, and it will consolidate the memory. Consolidation of memory mean to convert the memory into solid form. I'm going to show you how it will convert the memory into solid form. Memory into solid form, then or to a structural base. If we give a structure to the memory, then that memory is already converted into long-term memory, and that memory cannot be deleted. That memory exists in the brain up to lifetime. So how it the second messenger formed over here will be transported toward the cell body. Inside the cell body of this neuron, a special type of protein is there that is called CREB protein, CREB protein. You can call it CAMP response binding protein. CREB, CREB, CREB protein, or CAMP response binding protein. So this CAMP response binding protein is very important protein because the with this protein the CAMP will bind. The CAMP will bind with the CREB protein. So this is called CAMP response, RE for response and B for binding. Then protein is protein. So the by combining the CAMP with the CREB protein, the CREB protein will bind with the CREB protein will be here, and the CREB protein will bind with the CAMP. After binding with the CAMP, it will form an element that is called CRIB element, or CAMP binding protein, or CAMP binding element, that is called CRIB element. This CRIB element is actually very important. This CRIB element will CRIB element is actually a combination of protein and CAMP. So this CRIB element will be transported towards the nucleus. This CRIB element will go inside the nucleus, and it will combine with the it will activate this transcription factor. So the transcription factor will activate the DNA to for transcription. So the DNA will form what? The DNA will form messenger RNA. So the messenger RNA will come out of the nucleus, and that messenger RNA will start what? Start the synthesis of protein. That protein is actually synthesized for the formation of structure to the memory. I'm going to show you. That protein will do what? That protein will be transported towards the periphery of this of this neuron. That may be transported toward this side, that may be transported toward this side. It depends upon the memory, or it vary from memory to memory. So what will happen? It will, for example, this memory is transported toward, or this protein is transported toward this side. In the previous picture there was no dendrite over here, so a new dendrite is formed in this one. So this dendrite represent an engram. So you can say a small part of this dendrite, a small part of a small section of this dendrite may be engram, or this whole part of, or this whole dendrite may represent an engram. So this engram, or this newly formed structure, represent what? This represent the new memory. This represent the memory which is stored by the memory which is stored by the neuron, which is converted into solid form. So a very mature neuron will contain large number of dendrites. It means that the mature neuron will contain large memory. Now, if we are increasing in age, and we are storing memory from day to day in daily life. So what happened? The number of dendrites and number of nerve ending is going to increase day by day. So why this happened? This happened for the storage of memory, for the storage of information, for the consolidation of memory. This is called what? This is called consolidation of memory. We All types of memory cannot be converted into solid form. All type of memory cannot be consolidated. Only the long-term memory is consolidated. once the memory is consolidated, once the memory is converted into solid form, that memory can be recalled. That memory can be recalled, and how that memory can be recalled? For example, this memory is stored in this area. Now, if we want to recall this memory, we will have an impulse towards this area. When we will get the impulse from this nerve ending, and that impulse will goes as action potential toward this engram, toward this nerve terminal. So if the action potential goes to a special nerve terminal, that will show a specific memory. Now, for example, this nerve terminal will show a different memory. This nerve terminal will show a different memory, and this nerve terminal will also show a different memory. Each and every part of this neuron inside any memory site, inside the hippocampus or inside the amygdala or the prefrontal cortex, will show a specific memory. So that specific part for the designation of special memory, that is called engram. For example, this area may be an engram. For example, this whole area may be an engram. For example, this small part can be an